So the first half hour that we were assigned, or whatever time we take, is on delivery. So we start with the challenge of therapeutics delivery. And so just to set the stage in just a couple of words, as you probably all are aware, the collagen 6 muscular dystrophies are a special case amongst muscular dystrophies, in that the primary genetic defect, if you will, or the primary genetic problem is not in the ready-made muscle cells. So it is in cells that are supporting muscle and create the connective tissue environment of muscle. And these are a particular cell type that's being defined at the moment. They are fibroblastic cells, but they're really more mesenchymal stem cell-like cells. They're basically cells that have a mission to create matrices and extracellular environments that support the muscle, both structurally, but also by signaling, providing growth factors, signaling factors, interacting with receptors, controlling mechanisms within the muscle cell. And so the unique situation that we have here is that we have a predominantly muscle disease that is not caused by primary defect in muscle, but it's caused by a problem of the muscle support system. So the cells that are important are these cells that create these matrices around muscle, as well as probably also the, as Paolo Bonaldo pointed out, also the stem cell population of muscle itself, which are the satellite cells. In addition to that, we have an additional challenge in that, as you probably know from your own families, collagen 6 disorders are not just a pure muscle disorder. They also affect other connective tissues, sometimes more obviously, such as the skin, with different texture of the skin, different formation of healing of the skin, but also probably tendons and joint capsules with the contractures and the hyperlaxity that we see, the joint shortening that we see, and maybe even some of the internal organs. Sometimes we see bladder problems, sometimes vessel problems, blood vessel problems, and probably some other things that we haven't really fully understood. And so that plays, of course, into the consideration of delivery of a therapy. You want to deliver a therapy to every cell type that needs it, and not leave anyone out. So the current tools in neuromuscular genetics therapies are, of course, focused on muscle, because most of the muscle disorders affect muscle itself. And so in the collagen 6 field, we have to take a step back and say, well, how do we get actually into our cells of interest? So that's the background, that's the challenge. And so now we're going to have perhaps, we have two experts sitting right here, and they're going to tell us about how they're going to address these challenges.
Okay, good afternoon, everyone. First, I would like to present. I am Arístides López-Márquez. I am a biologist. I am a molecular genetics, and I'm working in the Sant Joan de Déu Hospital in Barcelona, in Spain. Okay, Sant Joan de Déu Hospital is a pediatric hospital, maybe one of the most important pediatric hospital in Spain and also in Europe. In my case, in my lab, under the direction of Dr. Cecilia Jiménez Mallebrera, we are working also in collagen 6 related disorders. And now we are working in two strategies, nucleic acids based therapies. For example, on the one hand, with CRISPR-Cas, in order to correct dominant negative mutation in COL6I1, and also to try to specific silencing of the mutant allele in this case. And on the other hand, we are also working with antisense oligonucleotides that are small nucleic acids sequences to try to also silencing the mutant allele in the fibroblast-derived patient fibroblast. Okay, so at this.
What the nucleic acids are.
Okay. Okay. So we have checked, we have showed that these strategies are working in vitro, and now, as Professor Bonnemann said, we are also worried about the delivery strategies, how we can take all these potential therapies to the fibroblast in an in vivo model. For this reason, we have generated also a mouse model with the specific mutation, and now we are checking that this model is also presenting the pathology. And we are also working in a new strategy with nanovesicles that are based on lipids, are like small droplets made of lipids, in order to put this CRISPR-Cas machinery or the artisans and oligonucleotides inside the droplets in order to try to give the cells these strategies. So at this point, we are working in these 2 or 3 strategies. I take the opportunity to thank CureCMD for the nice invitation to be here. It's my first occasion, my first opportunity to share these 3 days with families, patients, other colleagues, excellent scientists, and I am learning a lot. And I will come back to Spain full of energy to continue working in this strategy. Thank you.
Now let's just maybe discuss this.
Yes, yes.
So just to reiterate in just my own words and other words, what this technology is about. So this is a technology where you use, actually, most of you probably have been exposed to this technology because that's how the COVID vaccines are encapsulated. So they're basically put into lipid droplets, meaning fat droplets, that are encapsulating whatever you want to deliver in a little droplet, and then you can inject it. So in the case of the COVID vaccine, it's directly injected into the muscle, of course, and then taken up by the muscle cells, and they make the COVID part that the immune system needs to recognize. But here the challenge is, of course, to make similar particles. There are thousands of variations of these that can go by injection systemically to all muscles where it's needed, and then into the fibroblasts in the muscles where it's needed. So these are not viruses. They're not recognized by the immune system as foreign, they're inert. They have a different, you can actually make them as big as you want and probably load a lot of stuff into them. So those are the advantages. And what are the challenges?
In my opinion, the challenges are how we can direct our nanoparticle, our nanovesicles to the fibroblast, specifically to the fibroblast, not to other cell types. In this topic, maybe we are thinking about to combine, to mix this nanoparticle with other molecules that drive specifically the nanoparticle to the fibroblast. It's a real challenge, a really important challenge. It will be our future projects for sure.
Mm-hmm. All right, let's just take some questions here.
Just for everybody that wants to ask questions, please, if you want to raise your hand, that's fine right now, but I'd like everybody to use the app. So please go to meet.ps/col6, and then we can have them queued up. We have some left over from this morning. I don't know if they're relevant or not. There was a question about, is head lag seen in collagen 6 or 12? Somebody was asking about just head lag being in collagen 6 or collagen 12.
Oh, yeah.
That's really probably more clinical.
That's maybe a more clinical question that we can maybe address later. And also other scientists in the rooms, please kind of speak up as well, how you see the advantages or challenges of this approach.
Dr. Bonnemann, I do have a question that just came up as well. So they want to know, are we still looking at AAV or is this lipid text barriers?
Are we talking about AAV now or after?
I'm just trying to see whether any questions for clarifications are out there about the nanoparticles, the technology, any comments?
I had a question. So you're combining different type of lipids, right? And are you also mixing proteins?
Microphone.
Are you asking maybe for the specific delivery? No, at this point we are performing the ratios between how many nanoparticles we have to put in the mix and how many nucleic acid-specific therapies, for example, antisense oligonucleotides or CRISPR-Cas. When we check that this strategy is working in vitro cells in the laboratory and also in the mouse model, then we will move to the next step in order to put these proteins. We are thinking already about which protein we can use to increase the specific delivery to the fibroblasts in the muscle. But I cannot say how many proteins now.
And here, of course, the first challenge raises its head, and as soon as you introduce proteins into your delivery vehicle, the immune system takes notice, and so you have to be very careful about the immune consequences of that. So there's a question in the chat that leads us right to Veronique, and which is, are we now all lipid nanoparticles, or is AAV any good here? Do we have to look at AAV? So again, one sentence of introduction. AAV is adeno-associated virus, which is a non-pathogenic virus that in its own right can't even exist or replicate. It always needs bigger viruses to take it along for the ride. That's why it's called adenovirus associated virus. So it goes with the, for instance, an adenovirus, because other viruses deliver parts of the genome that the AAV itself is missing for replication. So it's really the virus that's harmless that's out there in the population. And unfortunately, also a number of you in the room have already seen this virus and in its various iterations and made antibodies against it, which is a problem, of course, for delivery. The concept of gene therapy, however, is to gut the virus, to take out the virus genes that the virus needs to replicate and cause an infection, and replace it with genes or nucleic acid that you want to deliver. So basically, all that's left over is the envelope of the virus, of the AAV virus. The envelope is made of proteins, and it is the part that the virus needs to infect cells. So a normal virus infects the cells by being recognized by the cell surface on its envelope, and then gets gobbled up by the cell and then delivers the genes. And a pathogenic virus or a virus that wants to replicate then takes charge of the cell machinery and makes it do more virus replication. And in the gene therapy, that's no longer in the virus. In the gene therapy, there's now the delivery that you want to achieve. There is a gene in there that you want to have expressed, for instance, in the muscle. And you create that in bioreactors. You basically create in one set of experiments, you create the capsid, and then you fill it with the cargo that you want to deliver. So it's really a delivery tool. However, the cells it infects are dependent on the envelope, on the serotype. And here we have a problem.
Yes, so, well, you may have heard several natural occurring serotypes that are commonly used, for example, AV8 or AV9 that are used in other clinical trials. So we've been working with AAVs for delivery to Fabs for at least 8 years now, but our strategy at first was to look at a deliver AAV that expresses a fluorescent protein. And I think we were underpowered because when these AAV enter in the muscle fiber do take up these viruses, and they express the fluorescent protein at high level. So it was masking whatever fibroblasts could express. So now we're switching strategy. We are still screening for naturally occurring serotypes, but we're using a genetic strategy where the AAV will change the DNA inside the fibroblasts and allows the fluorescence to be expressed inside the fibroblasts, and so that it will be a higher and stable expression that we'll be able to detect.
In simple terms, but if the virus gets into the right cell, into the fibroblast, it actually flips a light switch and turns on the light in the fibroblast. And you can see it. And when the fibroblast lights up, you know the virus got in there.
Yeah. So I think with this approach will be much more successful at carefully comparing the different serotypes and determining which one is best for FAPS. And then we are also looking at novel serotypes. So we found a collaborator in Boston, Dr. Luc Vanderberg. He's created a library of viruses that are based on the phylogeny of the naturally occurring serotypes. So basically, they're kind of the ancestors of the AAV that we see now. And so each of these serotypes is barcoded. So we're using those barcodes, the presence of the barcodes in the fibroblasts to determine which one is efficient at transducing the FAPs.
Veronique, do you want to introduce yourself just briefly?
Oh, my name is Veronique. I'm originally from Canada. I came to Carsten's lab 10 years ago, and I've been working on developing molecular therapy for collagen 6 since then.
So the opportunities are AAV is a workhorse in gene therapy. So there's lots known about AAV. We know how to produce it. We know how to purify it. We know the analytics of it. And you can put anything in there that's a nucleic acid or DNA that you want to deliver. However, it has an envelope size limit. It is a small virus, so it only has a capacity to a certain point. So for instance, if you wanted to replace the collagen 6 genes, you could do that with A1 and A2. They're small enough. Can't do it with A3, too big. So that's where that falls apart. But you can deliver other things as well. You can deliver a CRISPR technology. You can deliver a gene regulation technology, knockdown technologies. It's a good delivery tool around all of that. Problems are if you use natural serotypes. Some of us, maybe 30% at school age, are immune already against some of the serotypes, and then you can't give it because the immune system will just take it away. It doesn't cause any harm. It just kind of will be inactivated and goes away, not efficient. Second problem is if you can get it, you have no antibodies. After you've gotten it, you have antibodies. It is basically a vaccination that happens at the same time. You can only give it once unless you do more sophisticated tricks around it. And then one has to be very careful that one doesn't irritate the immune system by too high a dose. And that has happened in clinical gene therapy in Duchenne muscular dystrophy and others where a very high dose can cause immunological problems just from the load of virus that you give. And so the art is to give it as low as possible, but as efficiently to the cell where it's needed as possible. So that's where that's heading. Questions, comments?
So, Dr. Bonnemann, there's a lot of questions in general talking about, timelines. What does this look like getting to, the therapeutics, obstacles you all run into?
Save those for the general discussion.
General discussion. Okay.
Any questions about AAV versus nanoparticles?
There is an AAV question about, will it help with exon skipping or a different strategy completely?
Yeah, so AAV can be, Jeanette, you want to?
Yeah, we can actually expresses the antisense sequence with a U7 promoter, a specific promoter, and it will act just like a antisense oligo, just like if we were injecting the naked antisense oligo, but it will be expressed continuously.
Yeah, so you can deliver a lot of different tools with that, including an exon skipping tool or a mutation knockdown tool, if you engineer it right. So those were the delivery options for larger payloads. There's also, if you want to give just for some of the applications that we're going to talk next, you can get away with a small nucleic acid that you want to give, a small ASO or a small RNA to influence the mutation. And those need to be delivered to the fibroblasts as well. And there are similar principles apply. We have to find a chemistry that delivers properly to the fibroblasts. And there are also several tricks are in play, including lipid conjugation, kind of adding a little lipid tail to the nucleic acid to make it go to the fibroblast. Is there no other questions specific to delivery, AAV nanoparticles? Then we can go to the next part is why do we want to do that? What do we want to deliver?
Can I just add one last thing? We should mention, because we're focusing on muscle FAPs, but as Carsten said, This is just a start. Of course, we will look at tenocytes and other organs.
Yes,
For-
other
yeah,
connective
mhm.
tissue types. Yes, that's what I was.
Dr.
But-
Bonnemann, what about with nanotechnologies, both dominant and recessive? Does it matter?
Yeah, so again, then the nanospheres have the same advantage of being just a tool to deliver all kinds of different things that you can use to influence.
No, also I wanted to talk about that this nanoparticle. Okay. This nanoparticle has been already used for other pathologies, other pediatric pathologies, for example, some type of cancers.
And liver disease.
And liver disease for sure. And for this reason, we are really hopeful with this strategy. In my opinion, I think that these two strategies that we are talking about today, in parallel would be a good option for sure, because both of strategy has been already used for other pathologies, has been really increased, performed in the last years. I think so.
Yeah, no, exactly. And they're really tools to load with the specific genetic manipulator that you need to address the mutation. So that brings us to maybe part two of it. What do we need to deliver? And so there, just a high altitude view again, as you probably know, there are two different types of collagen 6 disorders. There are recessive ones where there's no collagen 6 or not enough collagen 6 being made because there are mutation or pathogenic variants on both copies that make a collagen 6 gene. You remember that every person has 2 copies of each gene, one inherited from each parent. And if both copies have a genetic change that doesn't allow it to make collagen, then you have a recessive deficiency of collagen. So there the trick would be, I just put in normal collagen and I'm all done. That is in principle the case. And so that will be an application for just gene replacement therapy. But as I mentioned before, COL6A1 and A2, yes, COL6A3, more difficult, it's bigger. And so we have to come up with other tricks, but that's where nanoparticles, for instance, could come in. They can take more cargo. The other genetic mechanism is the more common one in our parts of the world, which is dominant negative. So here, only one of the 2 copies of the gene carries a change, but that change is such that it interferes with the normal copy on the other gene and doesn't allow it to work properly. Matter of fact, if you only had 1 copy that works normally without any interference, that's fine. You would have no disease. But if you have one that is interfering, that's toxically interfering with the normal copy, you have a dominant negative situation and there is not enough functional collagen around to help the muscle. So here, the genetic strategies would be to either repair the copy that has the genetic change or knock it out so that it doesn't interfere anymore, or manipulate it in any other way that it cannot be visible to the normal copy of the collagen so that the normal copy can act independently of the mutant. So those are the strategies that one has to consider. And they are highly dependent on the mutation, or they can be more globally applicable in downregulating the bad copy. And we have examples for all of that in the room here assembled. Let's start with a point mutation, a single change in the collagen that causes one amino acid to flip into a different amino acid. And it's a very common occurrence, which is the glycine amino acid that's important for collagens change to something else. If that glycine is changing to something else, the chain will be acting as a toxic product and interfere with the normal change. So we have some strategies lined out to deal with that. So who wants to start?
Should we invite Astrid?
Astrid, come forward.
Astrid,
Francisca.
Francisca, yeah.
Yes.
Who is on Zoom? Oh, Jeanette.
Oh, Jeanette can talk about that. Yes, fantastic. Cool. Welcome, Jeanette. Can we bring up the Zoom?
In the back.
If anything I say makes no sense, please You know, we're under confidentiality, it won't get out. So you can ask any question.
I think there's like you said, when we get to the point, there's a lot of questions around timelines and,
Yeah,
you know,
we get to
translation
that.
and
We
helping to get
Let's
to
just
there.
lay the groundwork
I
here.
would like to only to remind that these strategies that we are working on in our labs, okay, for sure we are working maybe with one or two specific mutation, but they will be valid for all the mutation, no? Because if we try or we reach, for example, the optimization of nanoparticle or AA view, this delivery system will be useful for, okay, collagen 6, A1 and A2, for sure. A3 is more difficult than, as you said before, but it could be valid for all the cases.
Yeah, so as you will see from, it gets a little complex here now, but as what you will see is some of the strategies are mutation specific. They're designed to address one particular mutation, but the principle is portable to other mutations. So you have to kind of tune it for every single mutation. Another approach that Dr. Erdmann, Jeanette will describe and that Franziska reported on in our scientific session is to just find an off switch for that chromosome that carries, or that gene that carries a mutation. So you would just kind of say, well, I have two chromosomes, A and B. I know that B carries the mutation, I'm going to switch off B. Or you have chromosome A carrying the mutation, and I'm going to do for the different tool to switch off A. And then it doesn't matter which mutation is in A or B, you just need to switch the right one, A or B, off. So that would be a more universally applicable one. Okay, who wants to start? Oh, hey, Jeanette.
Hi. Can you hear me?
Yes.
Okay.
Maybe you
Good
want to
to see
start,
you.
Jeanette. So a brief introduction, please, of yourself, Jeanette. And then...
Yes. Okay. So my name is Jeanette. I'm a professor for cardiogenetics in Germany, in Lübeck. It's in the northern part of Germany. I diagnosed myself 10 years ago as a patient with collagen 6 CMD, and I'm carrying such a glycine mutation Carsten talked about just a few minutes ago. So 5 years ago, I decided actually to establish a working group at my institute to try to establish a therapy for collagen 6. And I'm totally happy that I can meet you virtually, and I'm extremely happy that Franziska and Nadine can join you in person. And so I hope next time I can meet with you.
Can you describe in 1 or 2 sentences, in simple terms, the strategy you're taking? That's challenging.
So actually we started using antisense RNA to suppress the mutant allele, the mutation, the gene copy that carries the mutation. And this works in the Petri dish. Actually, we can rescue the phenotype in our Petri dish. But then...
das habe ich online zu Exi Weekend Rescued Kinotime gefunden. Guck mal.
Sorry, it's...
I'm interference.
Yeah, Siri is interfering. I don't know why. But then Franziska came up with another idea. She read in a paper about, so we are now using CRISPR, and specifically CRISPR-off, to silence the mutated allele. And as Carsten pointed out before, so everyone who's carrying a glycine mutation, it's almost a private mutation. So there are rarely 2 or 3 or 10 patients carrying the exact same mutation. So if you think about therapy, it would be extremely expensive and difficult to design a therapy specifically for private mutation. And this is why we try to find a common variant that is, we say, in phase on the same chromosome with as the private mutation. And then we target not the private, the rare one variant, but the common one.
That's what I meant with this A and B. So always have two chromosomes, a chromosome A and a chromosome B. So Jeanette and her team are developing an off switch for A and for B. And what you have then figure out is my mutation on the A chromosome or on the B chromosome? If it's on the A chromosome, I use the A switch. And if it's on the B chromosome, I use the B switch, ideally. So yes, thank you. So let's go to Astrid.
Hello, my name is Astrid Brull, and I'm a postdoc in Carsten's lab at NIH. I'm working in actually two projects. The first one is the silencing of a specific glycine mutation. And another project is with RNA editing. So with this project, instead of trying to silence the mutant allele, we try to correct it. So changing the adenosine that is mutated and changing to the correct sequence.
So that's an approach that was developed only recently, where you actually, every cell has its own repair mechanisms, both for DNA, but also for RNA. And it uses this to kind of keep constantly occurring mutations at bay. And whenever you have genetic turnover, there's always mutations sneaking in, they always need to be repaired. And so every cell has a repair mechanism. And so what this technology does, it directs the repair mechanism to the mutation you want to correct. So it uses a special search function, a special search oligonucleotide RNA to direct the repair machinery. Hey, I'm here, do repair here. And then the repair machinery will be recruited to the mutation and corrected. So that's the base editing on RNA. That's another approach. And then we have a third type of mutation that only was discovered by colleagues and ourselves recently, which is a relatively common mutation. That's a deep intronic mutation that's in between the actual coding parts of the gene that causes an extra piece of nonsensical DNA to be made into protein and cause a toxic effect. So we call that a pseudoexon, an exon that doesn't belong there because of a deep intronic change. And here we have another technology to address that. Veronique, you want to just explain that?
So here we're using what we call exon skipping. What it means is that when the DNA is transcribed into RNA, this is when the pseudoexon drinks. Sorry, I have difficulty to do in lay terms. I was not prepared to explain. So basically, the RNA has to be spliced so that only exons are retained in the final messenger RNA. And this pseudoexon is a piece that gets inserted, okay? So after splicing, the pseudoexon is retained in the messenger RNA. So what we're trying is that during splicing, we're gonna mask with antisense oligonucleotides that will bind to the pseudoexon sequence. It's gonna mask and it's gonna prevent the splicing in of the pseudoexon. So at the end, we'll have a normal copy. So basically, it has high potential because it can basically correct the aberrant sequence.
And yeah, imagine a gene is a book, and the book contains a novel. But there's only certain chapters that are written in actual prose, and in between there's lots of garbage, random pages of random signs and letters. Now, if you were to open the book, you couldn't make sense of it. Only if you literally splice, cut out the prose, put it back together, and make a novel that makes sense by cutting all the garbage places out in the middle of it. That's splicing. That's how the genetic information is put together in something that the cell can understand and read and make a protein. And in a pseudoexon, one of these garbage pages now suddenly ends up in the novel and causes havoc in the flow of the text. And so this technology will prevent that splicing in to do that. So great. So let's talk about questions.
So a general one that I remember you talking about in Chicago, people with all these different types of variations we can have on the 3 different genes. Is there any correlation in severity between which gene is affected and any?
Yeah, that's a really good question. So there is there's some correlations. They're never 100% one-to-one. So there are some glycine, if we start with the glycine mutation, there are some locations where the glycine mutations are particularly nasty in being toxic on the assembly, and then the patient will be more severe. The same is true for the exon skipping mutations, where essentially a page that belongs into the novel is not included into the novel. So that's another type of mutation that we can address with this technology. And again, it depends on where that exactly happens, that skip of an exon, the severity. The pseudoexon mutations also tend to be more on the severe side, unfortunately, unless they're diluted out by normal cells. We call that mosaicism, that can also influence the phenotype. But the bottom line is, even if you have the identical mutation, the severity may be different. And that is because every human being is his or her own genetic unique assembly. And so some are more resistant and some are more vulnerable to these genetic disorders. And that explains some of the variability of severity that we see. So it's not all one-to-one written in the mutation. It also depends on the background where it occurs.
Another set of questions kind of get to, age of what ages could these therapies potentially be applied to, and questions about contractures, could they be eliminated or?
Wait, let's save the timing of the intervention and the timing of therapeutics development for the general discussion.
Yeah.
And let's talk about the third topic here, which is how we use cells to maybe think about therapies, and also use cells and model systems to look for things that are not genetic medicines, but that are medicine that could still influence and ameliorate the disease. So here we have Paolo. Oh, yeah.
I need respect. Okay, put you on the spot. Should I present myself?
Yes,
Okay.
please.
So I am a cell biologist at the University of Padova, and I've been working with collagen 6 since prehistory, since its discovery in the 80s. So actually, we produced the collagen 6 knockout mouse before there was any link with the human disorder at that time. And since collagen 6 is really a multitasking protein, it's really fascinating, this protein, the matrix, because it is quite complicated, as you understood from Carsten. There are 3 main genes, but there are also 3 additional genes. We don't know much about those additional genes, and there are many different parts of the protein. Is also expressed in different tissues. As Carsten was saying, it's very important the fact that it's not only muscle, but in muscle is produced by fibroblasts, or the fibroblast adipogenic precursor, this kind of mesenchymal cell, but it's also found in other tissues. And is also expressed during development in different organs. So this mouse model for us was a way to understand the function of collagen 6. And at that time, we were pretty surprised that the mice were alive and they were weak. And then we found that the mice had kind of myopathy. And this is how then later on was 96, the first link with Bethlehem myopathy, right? In 2001 with Ulrich. And this mouse for us became a kind of a model for the human disorder. Okay, we know we were being discussing this in the scientific part, the limitations of using animal models. Be careful that you cannot directly transfer what you see in mice in humans. And another point is that our mouse is completely missing collagen 6, not having kind of this different type of mutation. But anyway, for us, it was a way to understand the pathogenic processes downstream the primary genetic defect, the absence of collagen 6, what is happening. For us, it was a big surprise at the time, because the protein is an extracellular matrix, so this network outside the cells, outside the muscle fibers. And we found that there is a strong impact within the fibers. The protein is produced by fibroblasts and is deposited, is surrounding the muscle fibers like a network. And in its absence, we found that there is mitochondrial dysfunction. So mitochondria, there's the source of energy in muscle, very important, are dysfunctional. There is spontaneous apoptosis of muscle fibers, meaning that they are induced to die with the mechanism kind of programmed suicide of the cells. And later on, we found out that there is a defective function of the cleaning process of the cell that is called autophagy. This became important in the recent time for many different types of disorders, also in physiological conditions. And this is how our mice then became for us a way to understand whether there are targets for counteracting these defects. And indeed, is what we found in mice. There are targets. These defects are reversible. And we also studied the biopsies and culture from patients. We found that there are also mitochondrial dysfunction, defective autophagy. And this is how this brought us to another way to tackle the disease, not trying to correct the primary genetic defect, which is in theory the best way, you restore the normal collagen 6, but somehow counteract the downstream pathogenic process. And he had several nice results on this way. We had also, in collaboration with some clinicians, because we are cell biologists, carried out a couple of bioclinical trials that were successful for us. And we are still going on this, trying to find a way to counteract the downstream processes in mice. Yeah, right. And then there are different aspects to consider. One aspect that maybe Carsten was one of the reasons why you called me here, in the sense that you have to think not only muscle fibers are defective, was mentioned in the beginning, referring to us, there is also defective function of the muscle stem cells. It's called satellite cells, because surrounding muscle fibers, there are these small cells that are maintained there, usually in physiological condition. They are able somehow to restore fibers that are generating, but of course, in the myopathic dystrophic condition, the loss of muscle fibers is too much. And in this case, there is also a primary defect in the satellite cells. It's really surprising because these cells, they are producing their own collagen 6. And there's also this important aspect to consider. So there are different aspects, not only muscle fiber, not only satellite cells, but also tendons, because tendons also are important, of course, for muscle, and tendons are very rich of collagen 6. Yeah, I mean, it's a complicated protein playing different roles. So we should take care that not only muscle is the target for, to have a way to cure patient, but also we have to take care of other organs and get information that until now are not quite sparse. We are always focused on muscle. And Carsten said that, of course, not only mice, now we know there are not only nephrologic knockout mice, there are also other mouse models that have been produced with specific mutation, but then we have fish. Why fish? It's so far away from mammals or from humans, but fish has a big advantage because it's much cheaper, it's a faster development, it's transparent, so it allows also to have simple screens in fish. You have used fish, so we have produced collagen 6 knockout fish in order to carry out a kind of high throughput, large scale drug screening, let's say drug repurposing, using already FDA-approved drugs in order to see whether using simple screening fish, we are able to counteract some of these functional defects that we can detect in fish in a quantitative manner. So there are many different ways now in order to understand whether we find ways to know better the downstream pathogenic processes, the disease processes, and counteract them.
And the reason why that's important is because we can't put all eggs in one basket. Where a genetic disease is a difficult entity to treat, and none of these therapies that we're discussing here that go for the genes is ever going to be perfect or close to perfect. And so we have to make any effort we can to make the body more resistant to the defect of collagen 6. That's probably what nature does in some of the patients that have a severe mutation and end up being milder. They probably do some of these compensation mechanisms already because of their genetic background. And so these experiments will help to identify mechanisms and pathways where you can actually use a targeted drug to recreate this resistance against the different the missing of collagen cells. So all of these come together, then not mutually exclusive. They're really one big thing to follow. So let's get into the discussion of. And I said, do you have any specific questions around this? So let's go into the specific discussion. Now we're going to be challenged by.
I think the big ones are, what's this timeline look like? There's probably about 9 questions in here. At least half of them are around timeline. What can we do to help accelerate the research? What are your major roadblocks that you're running into? Do you see any mutations that are more, possible to hit first?
Can you bring up Dr. Erdmann, Jeanette, also, because we want to include her in the discussion. Okay, I don't have to talk all the time. If someone wants to say something, otherwise I talk. So, what are the challenges and timelines? All right, I take a step. So there are of course biological challenges as well as economic challenges to get to the goal of getting a therapy into patients. And so the biological challenges are still the ones that we'll work on most. So we want to be, as you can see here, there's different approaches, but we want to see which one actually has the most efficacy to go to where it needs to go, and does so in a way that we can expect that it will change the disease, and does so efficiently and does so safely. So that is kind of the major biological part that we have, but biological hurdle that we have to clear. It will perhaps initially be not a therapy that applies to all patients, because you can see there's such a variation in mutations and causation of the disease that we don't have a therapy unless it's one of those modifying therapies where they can apply across all. Is another advantage of them that could apply all across all subtypes. But if it comes to genetic therapies, we probably have to start somewhere and then use that knowledge to accelerate everything that comes after it. So after we've cleared the biological hurdle, then we have to see what actually cleared the biological hurdle. If it's something that's already been used in human, where we have a regulatory pathway, where other therapeutic agents of the same class have been used for other applications, then it's easier to get into the clinic, because we don't have to recreate every little bit of safety data, toxicology, and biodistribution. We have to do a lot of that, but not from absolutely scratch, and we can have more comfort from the uses in other application that is safe and applicable. That probably shortens the developmental timeline. How do we pay for that? So that is an unclear question. It depends on what you want to bring into the clinic. Is it something that's expensive, really expensive? Is it something that is cheaper? At the moment, I don't see that a big biotech player will just jump into that, because it's unfortunately too rare for commercial interest at this point in time, particularly if we go after selected mutations. But there are mechanisms now available both in Europe as well as in the United States, where you can apply for funding that's combined federal, industry, and philanthropic funding to insert yourself into a pipeline where many of the expensive parts will be supplied. So what's expensive? Toxicology is expensive, where you have to do it in hundreds of mice to just see whether it causes any organ damage in normal mice. And you have to do GMP manufacturing, meaning super high meticulous manufacturing with all kinds of safety checks at the end of it. Very, very expensive. And so these are the kind of multi $100,000 kind of roadblocks, sometimes $1 million roadblocks that you have to overcome. But there are now avenues to do that. There are grant mechanisms, philanthropic mechanisms to try to plug in if you come with convincing data. So if we have cleared those biological hurdles, have something that has a safety track record, that can accelerate the process. I hesitate to put years on it or a timeline, because the unexpected is always bound to happen. And then you sometimes have to take a step back or take a circumdeduction, if you will, to get to the goal. But it is close, yet far away. So objects may appear closer than when you have this thing in the mirror. Objects may be closer than they appear. No. Anyways, Jeanette.
Yeah, I can only second actually what you said. So actually, I have always a little bit difficulties to, yeah, to give more details on timelines or something, because I know that the expectations from the patients or the parents is extremely high. And I want to be honest, and I don't want to raise expectations what we maybe in the end cannot fulfill. So this is difficult. But on the other hand, I guess it's for me as a scientist, so actually I'm working in genetics since almost 30 years now, and we have now tools available to think about therapy. So this is really amazing. And so, yeah, it's on the one side, I think we can be optimistic, but on the other side, we have a lot of work to do and actually solve some problems we have not yet maybe even understood.
Yeah, that brings up a good point, which is the tools that are now available to us are completely different from what was available just 5 years ago. So there is a growing toolbox that is good and problematic. So good is you have every half year a new cool tool comes out, and you want to try that and see whether it works. But that always puts you back to square one, where you have to kind of start new and see whether that tool does it. And at some point you have to kind of decide where this is something that I see really possible to go forward to the clinic. And at that point where you kind of go that route, you stick to it. You can't just say midway, oh, I'm going to do something that's much more cool. Then you're back to square one. So at some point you have to decide, well, this is good enough, it looks promising, I'm going to go for that. And then that's a commitment from that point on that is not reversible. Then you have to go forward and do that. And that's why I said the more you can build on tools that others are using for other applications, the better it is, because then you can have more trust in that being a safe and valid tool that you can actually bring to patients. So that's kind of the compromise that you go for: coolness and state of the art and cutting edge versus something that has a track record and is already here in humans for other applications. So that's the kind of balance we try to take.
So obviously the question kind of also goes into people, what age groups would be best if and when these are available, right? It seems to be more effective with young versus old, and what impact does that have for contractures long term?
Mm-hmm. So here, that's again a question where I have to give non-committal answer, but I'll try my best. So we have to be aware, and there's no way to hide that the collagen 6 muscular dystrophies are muscular dystrophies. But by that I mean there are progressives. There are a degenerative disorder of muscle. Muscle will keep up with it only to a certain point and then get weaker over time. That's the nature of this disease. So that's why it's called a muscular dystrophy. That's all it means. Dystrophy means degenerative disorder. But muscle, on the other hand, and connective tissue are not tissues that are incapable of regeneration. Actually, they're quite good at regeneration, and if they have the opportunity to do so. And so in the collagen 6 disorders, there are, as Paolo mentioned, there are intrinsic problems with regeneration. So the satellite cells, the stem cells of muscle also need collagen 6 that don't have it. But maybe you can say, if I resupply collagen 6, maybe they come back to their senses and start regenerating better. Or it's a connective tissue that builds up, scar tissue that builds up in muscle, may be less irreversible than we think. We always say, well, once scar tissue is there, it never goes away. That may not be the case. The extracellular matrix is quite, and PhD expert on the extracellular matrix is sitting right here. And the extracellular matrix may be more modulable. There may be opportunities to kind of reform and rebuild the matrix in a better way if you repair the underlying problem. Nonetheless, it's fair to say that earlier is almost certainly better in this disorder as in any progressive disorder. But it doesn't mean that there's a point at which it is definitely too late. I don't think I want to put that in the room here. I think there's always opportunities to recover something if we get efficiently to the right cells. And that also is true for the tendon and joint capsule cells. So that's why it's important to also get into those cell types to at least have a chance at changing that. If we don't target them, we'll never see whether our therapy is going to work on them. That's why we have to target them as well. So here's my hand waving, trying to get out of this. Anyone else want to comment on this?
Just a very short comment to what Carsten was highlighting and underline here. Matrix at the cellular matrix was for a long time just considered a kind of support for tissues, but now it's clear, and Collagen 6 is a clear example, that there is a kind of a platform, very highly dynamic platform for signals of the cells. And I think the complexity of this is due to the fact that Collagen 6 there in muscle is playing this complicated role as an extracellular matrix protein. It's different from other kind of proteins that are inside the cell or even outside the cell, because this is highly, I would say, multitasking protein, playing different roles. So this is also highlighted by the fact that, for instance, we don't know many things about collagen 6. Why? Patient that have mutation COL6A1, COL6A2 that is very similar to COL6A1 as a chain is not able to substitute for the other chain. Although the two proteins are similar, it means that there is something having to do with the, the requirement for having the three different chains. And two of these chains are very similar. Another aspect, many people consider collagen 6 kind of ubiquitous protein. It's not true, because if you, everybody mentioning this in the scientific part, if you check the promoter, so the regulatory region upstream or the coding region of collagen 6 is highly complicated, because it contains several elements, several sequences specifically required for the activation of proper activation, the expression of the gene in specific sites, not only muscle, including tendons, joints. So the comment is we should know more, much more about the basic biology, not because I am a cell biologist, but because this will allow us to, to improve also the understanding of the disease, and also in the perspective also to have other ways to think about therapies and cures. This is my view.
Yes, and I would only second that. And but at the same time, of course, we move to therapies while we not completely understand the disease. So that's something that happens. We have to do basically. We can't wait until we completely understand the disease, but we can use every therapeutic experiment that we do to better understand the disease. So even if we try a therapy, we'll learn from that trial of the therapy about the biology of the disease. So, for instance, Paolo brought this up in the scientific meeting. We don't even know how long it takes to turn over collagen 6 in the matrix. Let's say we correct it. How long is it going to take to replace the mutant collagen 6 with a better collagen 6 in the matrix? Is it days, months? We don't know that. No one has the data for that. And so that depends on actually doing it in models, but perhaps in humans at some point as well, to really understand that part better and then iteratively improve on our therapies. So it will be a give and take between therapeutics and basic science. What we don't want to do is do anything that's stupid and makes things worse or causes a risk to the patients.
Any other comments from the Dr. Bonnemann? I do want to, there's a collagen 12 family here as well. Is there any carryover from collagen 6 to collagen 12?
Yeah, so are there collagen 6 questions that are still out there? So collagen 12 is another collagen that's out there in the matrix. It is somewhat different from our understanding. We don't know so very much about it from collagen 6 in that it isn't quite as hugging the muscle fiber as closely as collagen 6. It is probably more in the actual mechanical matrix outside of the muscle fiber. So if you look, if the muscle fiber is here, collagen 6 really hugs it. It kind of like a lacy glow. It goes really into it. And collagen 12 is an outer covering that doesn't directly interact with muscle, but it regulates tensile strengths in the muscle itself, but also in the tendons. So if you don't have collagen 6, everything gets hyper elastic, if you will. And you can imagine if you have a rubber band that's attached to a bone, if it's a relatively tight band, you can actually move the bone without much pulling. But if it's very elastic, hyper elastic, you pull and pull and pull and pull, nothing moves, and then at the end it will move. So in the end, it also causes weakness and unloading of the muscle, which then has negative effects on the muscle. So it's also a muscle connective tissue overlap disease, but it's more in the connective tissue than it is in the muscle, and collagen 6 is more in the muscle as a disease driver and less than that. So that's kind of the basics of that.
This opens also the view to the fact that not only collagen 6 that is so important for muscle, but maybe other collagens are also important for muscle, because, for instance, Ehlers-Danlos is a disorder of connective tissue, but there are patients with Ehlers-Danlos that they show some myopathy. These are other collagens, meaning that, again, matrix, as Carsten said, you need to have, it's highly dynamic, the proper interaction between this protein. We don't know much about these things. And it's clear that SciFam is not stable. It's highly dynamic. There are some results showing that even there is a circadian regulation of collagen synthesis, meaning that there is a continuous, these small changes in the matrix. And this is an aspect we don't know much about it.
Which again could work in our favor if we repair the matrix. It could kind of continue to turn over in a positive way. And so, I open it up for any questions.
There was a collagen 6 question I missed. Somebody asked about metformin. Has that been used in collagen 6 at all to prevent cell death? That would...
Not to my knowledge. I don't know whether it's been tried in any animal model. It's been tried in a lot of diseases, and so, but I have no data on collagen 6. Metformin. No, we don't have any data. So room, speak up. You sit here. Yes. Please.
Yeah. In the past, we've talked a lot about animal models for Col6, and I know that it sounds like there's been a fair amount of progress on that over the last couple of years. Could you maybe help everyone understand a little bit how far?
So animal models, so the first one was, of course, Paolo Bonaldo's animal model, and then now a number of other animal models. There's a collagen alpha 2 knockout mouse, there's a collagen alpha 2 stop codon mouse. There is a dominant model of exon skipping in Col6A3. There's a Col6A3 hypomorph, so making less than normal collagen 6. There is a pseudoexon mouse now that has a human pseudoexon that we created in the lab. There's a glycine mouse now. So these models, there's no fundamental difference in the disease understanding in them, but they do provide models for mutation-specific engagement of it. So the glycine mouse will help to engage the glycine mutation, the skipping mouse, the exon skipping, the pseudoexon mouse, the exon skipping. So they all serve their purpose. None of these mice is the human disease. That's very important to understand. So it replicates certain aspects of it, mechanisms, matrix composition, but it is not the human disease. And I think, and that's not unique to collagen 6. Many animal models in neuromuscular disease are imperfect. Animals are different species. They're different mechanics, they're different size, they have different turnovers, different lifespans. So many things, different regenerative capability of muscle. Many things are different. So I think it's important to ask the right question of each model, not ask the mouse to be the human disease. That's and not explain to the regulators, this is the human disease. Just ask the right questions that you want to ask. Am I engaging a pathway that I want to engage? Am I engaging a mutation that I want to engage? Do I have the right biodistribution? I think those are the questions that we want to answer from them. And so they're all valuable, but they serve their own purposes.
Actually, we had
also introduce yourself.
Okay, my name is Onnes. We are from Turkey. I just want to ask about drug repurposing. We haven't talked about it too much. This station, is there any progress?
And maybe Paolo is the best person to answer this because the zebrafish, the sushi model. The zebrafish model, he used it in the lab. Chiara, I think, did the work, sitting right here, to to use a library of drugs that is ripe for repurposing. And so maybe Paolo can.
Well, this was an idea we had of not relying upon this treasure mouse, because for us, really a gold mine, having found all these different pathogeny different in the mouse, but also moving to a simpler model. And since zebrafish has several advantages, that, you know, as Carsten said, you know that is not the human disease, and you know what to do with that model. So what we wanted to do with zebrafish is using zebrafish as a way to have the possibility to do a large-scale screening of already approved drugs. So what Chiara did actually was to produce again a knockout model in zebrafish. First, you had to characterize very well that the model is proper, and this is what they have done. Now, this work we are going to publish soon about the characterization of the zebrafish model. And then she was in a quite simple way, because it's not really expensive. We bought the library, we have these zebrafish embryos. You can test them at early stages, put in this called multi-well plates, so you can test many of them. And in each well, you can test a different drug and have a kind of quantitative output of measuring their movement. We have carried out this at 2 different times with the 2 different ways, and we have screened until now 300 compounds, and we found some nice results. Why we have done this? Since we already know we have drugs against to counteract mitochondrial dysfunction, we have some drugs able or therapies able to induce autophagy. Because we think the more we know about drugs able to somehow ameliorate any part of these defects, the better is. Because maybe you can think about a way to have already approved drugs or nutraceuticals, as we have also done with autophagy, and use them in combinatory manner. And we have nice results about it. So I think this is also a good opportunity to find a way to counteract the major defects, and a single drug maybe is not able to do that. So if you have multiple drugs and able to use a low level, not high level, that might also give some kind of unwanted effects, and use it in a combinatory manner, this might be the way. And we are doing this in fish. Of course, once we have found these drugs are working in fish, then we can move on mice, and we have the cells of patients where we can test them.
Yeah, exactly. So that's a screening platform, and fish is fish, and so maybe fish has its own collagen 6 problem that humans don't have, and you fix it with a drug, and then you have to validate. Of course, the drug is now highlighted on the screen, and then you take it and then put it to the ringers to see whether it actually would work in a human system. But it's screening is the purpose of screening is to highlight something that you haven't seen before. So that. That's the, and then if something comes out of that, then one could say, well, now it's validated across the system, and now we can repurpose it for the disease, because it's already an FDA-approved drug, but the FDA will treat it as a novel drug because it's a novel indication. So you have to still have to do a clinical trial. You can't just go, well, you can, but you shouldn't just go into the pharmacy and just kind of buy it off the shelf and eat it. Because you have to be sure that it doesn't cause its own problems in this specific disease context. So.
We got a couple questions that came through, and then we're about to get close to the end of the time here. So one thing I do want to mention is that Rachel will take a look at any of the unanswered questions, because we can get to all of them and try and get some responses back to people within the groups. And there's a lot of people been asking how you can help. And tomorrow morning, I'm going to plug my own talk, right? We're going to talk about Cure CMD and what our plans are and how we're trying to advance a lot of stuff tomorrow morning at 8:30. So please, you can hear a lot about where we're planning on going. But I think this is a kind of a good ending question for you. It's, been 3 years since we've had a chance to meet as a big group. What are some of the most surprising things that you've learned in that last 3 years that in the advancement of this research?
Okay, I'll pass those questions on. Maybe I have Jeanette start. Okay, I will start.
Sorry.
That's great.
Yeah, sorry, I have a bit of a trouble with my cat. Hey,
Let's have
sorry.
a cat comment on
So
this.
I don't know. So
The cat wants attention.
This is great.
yeah. Oh man. Yeah, he's very, to excuse him, he's very young. And yeah. Sorry. Good. So, Carsten, I didn't get your question, actually. I was a little bit distracted.
The question was, in the last 3 years, what was the most kind of either surprising or encouraging development that you've seen in relevant to collagen 6?
Huh. Actually,
Hmm.
I must say that my hope is higher because I see all the developments in other diseases like SMA or Duchenne, or for instance, I have a very close colleague from Hamburg, and she developed a therapy for hypertrophic cardiomyopathy for very small children with a really severe phenotype. And she managed now to get in contact with a company, and they are going to first clinical trials now. So this is something that gives me hope that someday we can also go this way.
Yeah, exactly. And I would have said very similar things, which is that in the last 3 years, the two things have happened. Many things have moved into the clinic in kind of gene and transcript directed therapies with sometimes surprisingly good outcomes, but sometimes also new risk profiles that emerge. But the entire field is starting to get a handle on how to do this and how to move forward. Again, this is collagen 6 disorders, but we're part of a much, much larger community of neuromuscular and genetic disorders. And the entire advancement of the field is cross-fertilizing other subgroups. So wherever a breakthrough is achieved, it actually pays off for us as well. And that's the one thing. And the other thing is really the explosion of molecular tools. I find that really astounding that we're now able to repair mutations ever more efficiently. I mean, at every couple of months, a new iteration of a technology comes out. So it's hard to keep up with. That causes the risk that you always want to be at the very cutting edge and then drop everything else and go to the next one and never get anything done. But it is really the speed with which this is now filling out, including some that are being dropped because all of a sudden, it's clear it isn't as efficient as we thought. It's not reproducible, and then you move on. But because these tools are tools that are applicable across so many different disorders, wherever they pop up and are tested, can be used to influence the research that we use specific for this disease. And I think the speed with which that is evolving has surprised me over the last years.
Well, more from the lab perspective, I think in the past 3 years, the development we've made with the mouse model, the pseudoexon mouse model, I think was very encouraging because we shown that the pseudoexon is expressed in this mouse, and not only that, the protein containing the pseudoexon peptide is present, so that validates this model.
Yeah, and the other thing that is worth pointing out for this model now has an entire region of this collagen 6 gene be completely human. So what Veronique did is she dropped in the human gene into the mouse, that part where all the mutations are. And now we can use therapies in the mouse that we can directly translate to the human, because that part of the gene is identical to the human. And so that's
Yeah. So we made a lot of progress with this mouse, and we're finishing the characterization. Yes. Other encouraging parts? Well, yeah.
Well, yeah, especially in the project that I'm developing for RNA editing, it was surprising because I decided to choose that approach to use with glycine substitutions. And that paper was published in 2019. And since then, there is 4 other approaches that they have been developed. So in the past 2 years, it's like the things are moving so fast that you cannot even try the one that it was developed 2 years ago, that there is a new one, that you cannot keep going. The things, they are moving so fast.
Back in Chicago, we were not even thinking about RNA editing, and now we already have data on that.
Yeah.
No, I agree with Veronique and Astrid because, okay, I started to work in Collagen 6 3 years ago, more or less, but we were talking, having lunch, I think, no, that in these 3 years, a lot of papers, a lot of new technology, new approaches that I think that they are really, really hopeful. In my case, in the lab, we are really happy also with the mouse model. It's a different mouse model because we are doing a specific mutation in the exon 10 of the COL6A1. And we are observing, we are detecting that this mouse is also recreating, is showing some pathogenic characteristics in the muscle biopsies, also in the movement, etc. And on the other hand, I was really happy with the result with CRISPR-Cas. We have shown that it's possible to silence in a specific way the mutant allele without affecting the wild type allele, the correct allele. That it's really important because I don't want to silence both alleles for sure. So I think that I hope that in the future, in the near future, all these discoveries and all these improvements is going to be more and more close to the real clinical application, I think, and I hope so.
Well, I am complementary side of what they are doing. This is let's say, the gold standard approach to restore the normal allele, to silence the wrong allele, or try to find a way, in the case you have this recessive mutation of collagen 6, to produce again collagen 6. Our approach is complementary in the sense that we were somehow pushing that direction in finding this disease process that was very surprising, really. As I said, it was a matrix protein that is impinging on this faraway signaling within the muscle fibers and mitochondria, cell survival, and autophagy. So in our case, what do we think is the best way for us is to continue in this path and trying to find the best, the optimal regimen of the treatment in mice, and then transfer this, all starting from zebrafish in mice, and then moving to patient cells, that is able to restore at the best a certain defect. And then we have another drug, and then we can use it in a complementary way. Until now, we have not done this experiment yet because we want to be sure. When you think about autophagy, autophagy is now considered a kind of a longevity-inducing process, an important process, but we have to take care, because if you use too much autophagy, then you might have problems, and you might have the opposite of muscle atrophy. So we have to activate in a proper way. This is why we are screening for different drugs. So I think we are pretty close to have clinical trials with the different kind of approaches, the genetic one, the one based on downstream disease processes with drugs. I think the only problem really is that the disease is rare, so we don't have so many possibilities to test in patients like in other diseases like the Duchenne. I think Carsten, you say much more. You can say much more about this.
Yeah, no, it's true. I mean, we have to be then. We can't try everything that kind of looks vaguely promising, and but you know, it's a huge burden on the families as well to do a clinical trial. Those of you who have helped us with the omega pill trial know what it takes to participate in the clinical trial. It's no trivial matter for the families and the patients as well. And so it has to be our responsibility to be selective about it and only move forward when it's really promising, is safe, and is right for the picking at this moment. But the basic concept is right. And I mean, the cancer folks have done this for decades. They use more than one therapy. They use multimodal therapies to just kind of hit the problem from different angles. I think that's absolutely important in genetic disease as well. And unfortunately, we have a new generation of researchers going into this field as well. It's really fantastic.
All right, unfortunately, we're at the top of the hour here. I do want to definitely thank everybody for not only for your research, but for taking the time to come visit us today. So thank you very much for the information.